Well, thank you, Pam, and thank you, Kate, and the library, and of course, Carl, particular thanks to you and everyone who came out. So just a brief intro, which is that not every science writer gets to play in the science sandbox, but then Carl Zimmer is not every science writer. He's so influential among researchers in the field that Yale's Department of Molecular Biophysics and Biochemistry brought him on as faculty. If we detail all of his accomplishments, we won't have time left to talk with Carl. So I'm just going to say that he's an award-winning New York Times columnist and the author of 13 books about science, one of them, incidentally, titled A Planet of Viruses. So we're thrilled to have Carl Zimmer with us tonight to discuss his latest bestseller, She Has Her Mother's Laugh, The Powers, Perversions, and Potential of Heredity. And by the way, it's a great title. We can talk about how you came to that. So before we splash into science, I'll start us off with a literary question in honor of our library partners. So Carl, you keep the reader hooked with stories that engage emotionally. There's the medical suspense of Pearl Buck's daughter, Carol. There's a nasty paternity suit against Charlie Chaplin. And then there's the mystery of what happened to Tsar Nicholas II and Tsarina Alexandra, plus their 5 children after falling to Russian revolutionaries. And these are but 3 mini dramas in your Heredity epic. So was there a particular novel or narrative nonfiction that inspired you?
You know, it's interesting. I mean, I definitely wanted this book to have a to feel like you're reading a novel, or at least to dip into short stories, I guess, because heredity it's a scientific question, but it's also very much a very human experience, and it's maybe one of the most profound experiences that each of us has, and so. So I just looked around for cases where there was an amazing story that just happened to be sitting there. You look in the history of science, and you mentioned Pearl Buck. Pearl Buck's daughter suffered from a genetic disorder called PKU, which is a really important disease, kind of in the history of heredity. And I didn't know that Pearl Buck figured in the history of science that way. And I started reading about it and reading her experience, trying to take care of her daughter and not understanding at all the genetics of what was making her daughter unable to speak. And ultimately Pearl Buck had to have her institutionalized. And I just thought, this is just in and of itself, this is just such an amazing, really heartbreaking story. So I looked for those, definitely, like when I would zero in on one of these stories, like Charlie Chaplin, I would say, yeah, this is going in the book.
But was there a particular author that had inspired you, or?
I guess, I confess I was an English major in college. I'm not a scientist, but I read a lot of literature when I was in college, and one of the books that really stuck with me was Moby Dick, and maybe I should have sort of taken it as a sign that I found the sort of the nonfiction stuff on whale biology really fascinating, and like that as much as the fictional story. You would get people who would complain like, oh, you just skip over all that whale stuff and just get to the story about the people. So now that I've been writing for many years, writing nonfiction, I guess I'm appreciating Melville as the nonfiction writer as well as the novelist.
That's a good crucible. So you opened the book with a personal tale. Just to set the stage, there you are, you're about to become a dad for the first time, and you go to a genetic counselor. And this gets you thinking about heredity far beyond genes. So if we were to free associate with the word heredity, many of us here tonight would probably spout out genes. And you snap that link, not entirely, but you go, again, far beyond. And just for example, you talk about us having trillions of microbes living on and inside the body, most of which don't come from our parents. You talk about mitochondrial mitochondrial DNA, which hitches a ride in each of our cells and gets passed down, technology, culture, it goes on. So what are the things that might most surprise us about what constitutes heredity?
Well, you mentioned, how we all think about genes you know, what, so we think that sort of genes like must link us to our ancestors. I mean, after all, that's what they gave us, right? That's what we inherited from them. But it doesn't really work quite that way. And so, for example, if you go back, say, 10 generations to this, say, the 1600s, and if you could magically like sequence all the DNA of your ancestors, about half of them, you have no DNA from them at all. There's no genetic link that you have to them at all. And that just has to do with how genes work, how they get passed down from one generation to the next. Your DNA gets sort of chopped up and shuffled around and then recombined over and over and over again. And so that basically you're inheriting these segments of DNA from your ancestors. And there's just not enough room in your chromosomes for DNA from all your ancestors. So some people get lost. And so, they're still your ancestor, but you have no DNA from them. So I think that, that alone should force people to think like, well, why do I think of this person as my ancestor? What does that mean? And what connection do I really think I have with them? I don't have all the sort of definitive answers in this book, but I do find lots of ways to basically take the sort of the comfortable stories we tell ourselves and kind of try to blow them up.
Beautiful. So if we were to ask tonight's audience how many have had genetic testing, whether through 23andMe or Ancestry DNA or another service, I bet we'd get a good show of hands. So any caveats or concerns or words of wisdom to share with us?
Yeah, I write about this in the book, and I mean, each of us might decide to get our DNA tested just kind of out of curiosity, sort of a hobby, and that's fine. But we should also sort of, first of all, recognize that this is an amazing scientific moment. Previous generations could not do this at all. The human genome was sequenced in 2000, and over the course, it was finally sequenced for the first time in 2000 at the cost of about $2 billion. So now, you can get your genes tested for, $100. I don't know what the going rate is now. I've lost track. It's so cheap. So this is a kind of astonishing. And so, when we get the results, we should really take a look at them, really appreciating what they really can tell us and also what they can't tell us. So, it's not like you will, find, a gene that's going to tell you like what you're going to die of. I mean, that's not going to happen. You will get some results which might, when you read them, you might be kind of confused by them. You could like, oh, here is a gene that, raises the risk of Alzheimer's by a certain percentage. Well, what does that mean? What are you supposed to do with that? And if you don't have a gene that's a risk factor for Alzheimer's, does that mean you don't have to worry about Alzheimer's? Well, no, there are other ways to get Alzheimer's. So, So when we get these results, I think that they can be really valuable, but only if we really only engage with them in a serious way, not just sort of kind of rely on our assumptions about how we kind of think heredity works. And the same goes for the results that these tests sometimes give you for ancestry. You get a big map and it says like, here's where your ancestors from. And well, how do they know that? Well, actually, all they do is they compare your DNA to DNA from a sample of people from around the world. And these companies get, thousands or tens of thousands of people, and they kind of try to get an idea of, what a sequence of DNA from Ireland or from Senegal looks like. But this is a work in progress. This is science in pretty early stage. And so, your results might change. From one year to the next, you might discover that they've redrawn your map. And you got to be ready for that. And you have to acknowledge that you're part of the scientific process. You're not discovering something absolute about yourself.
So the comparative aspect you're talking about is such a perfect segue for my next question, which is, as racial issues rock our world, let's talk about some of the takeaways from your genome test. Long before you got your DNA sequenced, so did two volunteers, one from Nigeria, one from China, as you tell us about in the book, and quite a few of your variants show up in their genomes. So tell us what you found out about individual Z's genome sequence and what it says about skin color and race.
Sure. So while I was working on the book, I had the opportunity to get my genome sequenced. This is different than, say, getting a test from 23andMe. What 23andMe does is called genotyping. So basically, if you think of your DNA as a text made up of letters, genotyping will basically look at about 1 in 1,000 of those letters. And that's enough to tell you a lot about your genes and about your ancestry. But obviously, like if you can look at all the letters, about 3 billion of them, you can learn a lot more. So I got my genome sequenced and then had scientists sort of help me to make sense of it. And it was really fascinating and helped me to work on the book and to sort of talk about this revolution we're having in understanding the role of DNA and heredity. So, and some of these scientists, they had such a good time with it. They just, they analyzed my genome and they gave me a PowerPoint lecture about my own DNA, and they kept referring to me as individual Z, which was interesting. But there are people whose genomes have been studied very carefully, just as a way to get to understand, human genetic variation. And as you say, there was a one person from Nigeria whose genome was sort of picked out as kind of representative of sub-Saharan Africa. There was someone from China who was picked out as sort of a representative of East Asians. There are other people whose genomes have been very carefully studied. But what was really interesting was just that, most of our DNA is pretty much identical, way over 99% is identical. And then you have these little variants sort of sprinkled here and there. Maybe, 1 in 1,000 or 1 in a few hundred of these letters are different from person to person. But they add up. And so what's interesting is that, I was compared to someone from China, and we actually shared about 2.3 million variants in common. These are variants that are not found among everybody. The Nigerian person and me shared 1.8 million variants in common. All three of us shared 1.4 million variants in common. Like it's a kind of a complicated Venn diagram. And I think this really illustrates just the fact that any sort of like simple idea of race, where race is supposed to be like these populations of humans with these clear, distinct boundaries that you can see in their DNA, it's just a pure fiction. It's just a pure fiction. There's more genetic variation from person to person within a population than there are between populations. And this really gets to that. If you're looking at genetic variants, you can find a lot of the same variants, if you bump into someone in New York City, and if you're in the middle of a rainforest in Brazil, they're mixed around. And that's because we humans descend from a pretty small group of individuals not that long ago. And so so all those variants have been sort of mixed together from the beginning. We still are dealing with, this incredible burden of the legacy of race, or I should say racism, So, race matters, but it matters in how groups of people are treated and live, on, the conditions in which they're living. So, the fact that African Americans are being hit really hard by COVID-19 is not because, oh, they're of a different race and therefore they've inherited, a weakness for this disease. That's not the case. What they've inherited is a position in our society where they have vastly inferior medical care overall, which has really set them up for a fall in this case. And so getting confused about that can have really toxic consequences.
Yeah, it's such an important message, social construct. So here is a question from a participant. In the book, you talk a lot about eugenics. Oh, I'm so glad somebody's asking about this. And the negative implications of how science was used. Are you concerned that the wide availability of DNA testing may cause a rise of interest in eugenics? For example, my DNA says 99% Ashkenazic Jewish. Couldn't it be used again for stratifying people?
Well, we should definitely be very aware of the history of eugenics to see how scientists and could help sort of pervert scientific findings to lead to really terrible outcomes. There's no way of hiding from that. And it's not, and we might think of eugenics as being, a lot of people associate it solely with Nazi Germany. Obviously, they were rabid eugenicists, but they got a lot of their best ideas and inspiration from the United States. So, 100 years ago, many leading geneticists were eugenicists and would talk about, the need to have unfit people sterilized. And so I write about this in the book about how it was that heredity came to be sort of reduced to just genes, and then this ideology suddenly saw that the solution to society's problems was to just stop the bad genes from getting passed down to the next generation. And of course, it was always some other group who had those bad genes, the poor people, the Jews, the blacks. It was never the group in power who had those bad genes. So I don't think that simply having access to DNA testing just on its own is suddenly going to sort of create a new sort of eugenics policy in the government. I don't think that alone is going to do it. But certainly if there are people who sort of bring really wrong and dangerous concepts to those DNA tests, then yeah, sure, they can abuse them in all sorts of ways. They can say anybody with this particular gene, will not be accepted to college or something ridiculous like that. That's always going to be a danger, and so it is important to really appreciate the history, to understand why eugenics was not just evil, but bad science, and to make sure that we don't make those mistakes in the future.
Do you want to maybe just tell us maybe a word or two about some of the arch villains of the American, like there's Goddard and, maybe Davenport, and just a word or two?
Yeah. I mean, I do zero. I mean, there were I should say there were lots of eugenicists in the United States, and many of the most prominent scientists in the country were eugenicists, but I focused in on a couple of them. You mentioned Charles Davenport. Charles Davenport was actually a very very important geneticist. I mean, the science of genetics only started in 1900. I mean, that's when the name was coined, and scientists were rediscovering Mendel and finally starting to be able to explain some patterns of heredity in terms of what they knew about the biology of cells. So that was a big deal. And so Charles Davenport was this geneticist who he did a lot of early important work studying birds and other animals to document some of these patterns. And he thought, well, maybe I can find these patterns in humans as well. And he had the very sensible idea of looking for diseases that kind of follow the same basic rules as Mendel's law. And so these would be diseases where, if you inherited just one defective copy of a gene, you would get a disease. And so Huntington's disease is a really good example of this. And he went and looked at genealogies of families where there was Huntington's, and he showed, well, this is the pattern you'd expect if it was just one gene that influenced the disease, and you needed just to carry one disease-causing version, because you inherit two copies of each gene, one from each parent. So he was right, and the basics of what he said about Huntington's was totally correct, and was a really important step in the history of genetics. However, he thought there were all sorts of other things in humans that he could explain with this kind of simplistic view of genetics. For example, intelligence. And so he got in touch with a psychologist, Henry Goddard, who you mentioned. And he was in New Jersey at a school for the quote unquote feeble-minded. And these could be kids with all sorts of different conditions, Down syndrome, epilepsy, all sorts of things, including just kids who were just considered trouble. So Goddard was very systematic and wanted to sort of try to reduce them to some sort of simple scale. And he discovered intelligence testing. And so, he brought intelligence testing to the United States. So when our kids take IQ tests or even SAT tests, that's all because of him. So he was trying to be very systematic about analyzing these kids. And here comes Davenport saying, hey, do any of your students have like families where there are feeble-minded members of the family? Maybe we could trace that in the same way I've been tracing Huntington's disease. And so they went off and did all this research on the families of these students in New Jersey, and came up with what seemed like to them incredibly compelling examples that just one gene maybe was responsible for feeble-mindedness. Looking at it now, it's all kind of ridiculous, but Goddard became incredibly famous for this, and the U.S. government actually brought him to Ellis Island to check out the immigrants coming off the boats and to decide which of them were feeble-minded and which needed to be sent back. And Goddard would make these claims like, over half of all Jews, Russians, Italians that were coming off these boats were feeble-minded. These ideas in the early 1900s led to sterilization laws. This wasn't just like things that people were talking about. This led to real action. And tens of thousands of people were sterilized under state laws that were passed at the urging of people like Goddard and Davenport. So it's kind of astonishing if you haven't heard about this stuff before, but it's all there. Those stories are just all there to be told.
Yeah, it's amazing. Why don't we talk about a little different aspect of genetic manipulation, maybe a happier one. So we've been hearing a lot about CRISPR, for a while now. And since COVID, it's bounced back in the mainstream media. So maybe you'll break down for us a little bit about how gene editing technology works and what role it can play for finding a cure for COVID, and plague aside, what can we reasonably expect from the likes of CRISPR in treating illness linked to genetic mutations, say, for example, sickle cell anemia, and where are we with clinical applications here?
Right. So CRISPR is a way to edit genes. There have been other ways of manipulating DNA for the past few decades. But as I write in the book, CRISPR came along as this really remarkable way to potentially change DNA in very precise ways. So you're not just kind of going in and just blasting out a gene and hoping that you hit the right target. You can say, I want to change this one letter in this one gene, and you can do it. And this again has only like come about in the past few years, and so scientists are still sort of coming to terms with all the things that they can do with it. And so you know, one of the things you can do with it actually, it turns out that not only can you change genes, but you can sort of zero in and recognize genes with this technology. And so it's possible that this might be actually a way to look for the virus that causes COVID-19, so that people, there are scientists at Harvard, MIT, who are working on just, you know, a test where you might just, you know, spit into a tube, and these CRISPR molecules are basically just sort of zero in on certain virus genes, and if they found them, then it'll be kind of like a pregnancy test. Like you'd like put a little piece of paper in the solution, and there'd be a line, and you'd know that you had COVID. And this is still very experimental, but it could potentially work. So you know, that's just one of many applications that have popped up for using CRISPR. But certainly, you know, for the book, you know, one of the important possibilities of it is basically letting us grapple with heredity. So, you know, if somebody inherits a faulty gene, you know, we've talked about PKU, we have talked about Huntington's. Let's say sickle cell anemia, big problem for the African American community in particular, but also for other people as well. And that's caused when you inherit two faulty copies of a gene, and your blood doesn't make the proper shape of its hemoglobin, and so it leads to all sorts of problems with your blood. And you know, it's right now the best that can be done for people with sickle cell anemia is sort of just to manage the symptoms. But you know, people, you know, they suffer a lot. They go through a lot of pain, and many are, you know, are dying in their 30s or 40s just of all the complications. So so one idea would be this. You know, our blood cells, we generate new blood cells all the time. They come out of our bone marrow. And so what you could do is you could remove some of these stem cells, as they're called, from the bone marrow, and use CRISPR to change those genes so that now these progenitor cells that are going to produce red blood cells, they have genes that will make the right kind of hemoglobin. And it just takes a little change to fix it. So then you just put them back into people's bodies, back into their bones, and now they have a population of these stem cells that can keep replenishing a healthy supply of red blood cells. That's an idea. That's an idea that people didn't even think of until a few years ago. And right now, there are actually clinical trials going on right now with real people who are getting these cells put into their bodies with the hope that they are going to, you know, if it all works right, that they would actually be cured of sickle cell anemia. So, so that really shows you the incredible potential for this technology that lets us engage with heredity. You know, you don't have to just sort of resign yourself to the genes you have, but you know, that then raises all sorts of ethical issues. You know, you could just as easily change the genes in a fertilized egg. People have done it. I mean, it can be done. It's not that hard. And you might say like, well, we want to fix that gene, but while we're here, maybe we want to make another change to another gene. Pretty soon you're really in an ethical minefield. And right now we're all thinking about COVID, understandably, but this ethical challenge is going to be with us, I'd say, for the rest of our lives.
The next question also brings us into CRISPR. So here it is. When you think about historical scientific disciplines, such as phrenology, for example, scientific in quotation, which sought to sort people into groups, the efforts were used to quantify differences, whereas genome analysis really highlights the similarities. Do you feel that science themes that hold the most promise hold the most peril as well? And then CRISPR, for example, in parentheses.
Well, I think that, you raise a really good point. And, it's interesting to look back at something like phrenology, which was, trying to say something about people, essentially just based on the shape of their head. The idea was that different parts of the brain would have different sizes and shapes, and then that would affect the shape of the developing skull. And so you just needed to feel the bumps on people's heads to get an idea of what was going on in their brains. Now, that wasn't necessarily, in some ways, phrenology was saying like, oh, well, there's a range of shapes, but then at the same time, there was also sort of a racist version of phrenology, where you would see, these diagrams in these books in the mid-1800s, where they would say like, okay, this is a Caucasian person's head, and this is an African's head, and this is an Asian's head, as if like they were just totally fundamentally like archetypal, like there's like these 3 or 4 fundamentally different shapes. And they're not. In other words, there's just like incredible amounts of variation. And the same goes for skin color. You could, if you only like, I'm a pretty pale person. If you put me next to, say, somebody who's like a Dinka from Sudan, like obviously we're going to have very different skin tones. But even within Africa, like you can pretty much find a range of skin coloration going from that Dinka who is like, almost sort of charcoal black, to people who are, tan to quite pale, and that's just within Africa. So, so this is a big issue for scientists right now. Like, on the one hand, as you say, there's a lot that binds us together. 99.9% over that genetic similarity. But there are variations, and those variations tell us lots of things. I think if we just, sort of keep just bearing in mind how easy it is to impose ideology on those differences, then they're worth studying. They are, they can tell us all sorts of things about health and disease. They can tell us about our history. But we just have to make sure that we're not using those as an excuse to sort of justify one group over another.
I urge anybody to ask a question, but I'll hop in with another one of mine, which is, so let's talk about epigenetics. Or experiences that can switch genes on or off and change our heredity. So many of us probably know at this point something about epigenetics, but methylation is not a household word, even though it's a major biochemical process in pretty much every cell all the time. So talk to us about methylation patterns and how they might influence you if, say, your grandfather survived a famine.
Yeah, so I'm actually kind of amazed that epigenetics itself, the word, is so familiar to people. I mean, back when I would go and give talks with people in different places that weren't my office, So I'd give a talk, and then at the end of the talk, if I didn't mention epigenetics, like someone invariably would say, "What about epigenetics?" And I'd say, like, "Wow, it's amazing that you know about epigenetics," because really, like 20 years ago, as a science writer, like I was really interested in epigenetics, but no one knew what I was talking about. So for some reason, epigenetics has really like plunged into the popular consciousness, and so I think the appeal is that it does sort of stand in opposition to some of our ideas about genetics being fixed and rigid, that's a problematic idea. But basically, epigenetics, as you said, is sort of like our sort of overall knowledge of how the genes that we inherit actually get used in our bodies. So, every cell in your body has the same set of genes, basically, and yet, a cell in your brain and a cell in a muscle in your arm are using very different sets of genes. And so, in your brain, they're keeping a whole bunch of genes turned off, and in your muscle, they're keeping other genes turned off, and so that's good, and you need that sort of epigenetic code to be stable. So as as your cells divide, you want one kind of tissue to keep making the same kind of tissue. You would not want your brain cells to suddenly be acting like muscle cells. That would be bad. So what gives it that stability? What keeps certain genes off and allows certain genes to stay on? It seems like part of the story has to do with what you just mentioned, which is something called methylation. So if you think of DNA as being this long string of letters I mentioned, your cell has all these molecules that come along and read the message in each gene. So it just sort of lands on the gene and just goes along and reads it. Now, there are molecules called methyl groups. There are these little caps, and they can just sort of grab onto your DNA at certain points. And they can potentially prevent your cell from reading a gene. So the gene is there, but it can't be read. It's been turned off. And what's really amazing is that when cells divide, they not only copy their DNA, very often will make copies of those methyl patterns, so those methylation patterns. In other words, you got the methyl groups showing up at the same points on the DNA in these two new cells. We're talking thousands, potentially millions of spots, which is amazing. So you can think of this as another kind of heredity. In other words, those cells are inheriting not just the same genes, but some of the same methylation. And so the big question that really excites people is whether that kind of epigenetic pattern could get passed down from parents to children. If you're a plant, if you're a tulip, yes, definitely that can happen. Plants have a lot of epigenetic inheritance, and that has to do with sort of the way that plants reproduce. They have a different way of reproducing than we do, so it's easier for those things to get passed down. There's examples in little worms that scientists like to study that in animals that can happen too. So and you can even change those epigenetic patterns in a worm's life. Like if they undergo some stress, like they have to fight some bacteria, then they can pass down a sort of an epigenetic inheritance to their offspring that sort of prepares them for this kind of stress. The same is true for plants. So that's all fascinating, and I write about them in the book. Now, it's easy then to say like, oh, well, we must have that too, and that can explain all sorts of things. And you just have to you just have to slow your roll when it comes to this in science, because there are a lot of reasons to be skeptical that epigenetics can work at all in humans. So for example, when a sperm fertilizes an egg in our species, something really interesting happens. I told you about all those methyl groups on our DNA. Well, the sperm and the egg have these methyl groups on their DNA too. When they fertilize and the cell starts to divide, the embryo strips off all the methyl groups, just, and then puts them back on again. And, you know, it's not really clear why that's happening, but one thing is for sure, that it's hard to see how a change that, say, a father experienced in their epigenetics could then be reliably passed on to their offspring if the epigenetics is getting reset. Nevertheless, there are some really cool experiments on mice, for example, where it seems like some sort of epigenetic inheritance can happen. And these are experiments where you take a male mouse and the mouse goes through some experience like being stressed out. And then basically they do IVF. They take the sperm from the mouse and they fertilize an egg, you know, in a dish, and then they implant the egg in a female mouse. And then they can see that those mice seem to inherit something in terms of the epigenetics and the behavior of the mouse. Very controversial. And we, you know, we don't yet know, like, you know, if your grandparents, you know, were Holocaust survivors, or if people have ancestors who endure the trauma of slavery, we don't know if somehow that gets passed down through epigenetics through the generations. It's tempting to think that that is the explanation for lots of things, but we just don't know. So that's my long answer. Maybe not terribly satisfying, but you know, well, it's really important. I mean, this is really important research, but it's very easy, I've noticed, for people to just immediately jump, you know, and say from very tantalizing but small scale studies, just big sweeping universal sentences.
So how are you fixed for time, Carl? We have 3 more questions.
I'm good. I'm good.
Okay, so as a scientist, I have always found your writing does a great job of boiling down science for non-specialists. That said, I sometimes struggle to effectively speak to those who refute science with, quote, alternative facts, whether conspiracy theory or otherwise. What tools have you found effective in handling these kinds of conversations?
That's a great question. You know, I have been engaged with this issue really from the start as a science writer for coming on 30 years, I guess, because I initially really got into writing about evolution. And so immediately when, you know, I'm this young science writer working at Discover magazine, and I'm the only one getting like hate mail. And this is back when you actually got mail in the mail, you know, just letters. And you know, I had all this hostile mail showing up, and other people would just be, you know, writing about chemistry or astronomy and be like, wow, what's the problem? And the thing was that evolution just was a very, could be very disturbing and very threatening to some people, and they would want an alternative explanation that they felt more comfortable with. So, and, you know, I've seen this sort of the same pattern happen with climate change, with vaccines, with all sorts of things. And now even, you know, with a lot of what's going on with COVID-19, there's a lot of just not wanting to accept what the science is saying and to come up with these alternate views, as you say. I think, you know, the most important thing to do if you're going to be engaging with somebody is not to treat them like an idiot. You know, like it may make you really like, you may get very emotional if someone starts talking about how, oh, there's really no evidence this vaccine works or something like that. Might make you angry. And so you're going to take out your anger on that person. Well, if you had some idea that you could sort of really engage with somebody and you know, get them to understand the science, like you've already lost that opportunity. That's just a fact. So don't treat people like idiots. Try to understand, how they are seeing things. Some people really feel like, well, I can't trust anything. Like everyone's lying to me. I mean, you will see a lot of this sort of conspiratorial thinking. Like the flat Earth people. Like they really feel like, oh, I can't trust anything, but I can trust something on YouTube. So like, well, why is that? Like what's your basis for accepting information or not? And you can start engaging with people that way. Explain how scientists know what they know. Don't just say like, well, this is what the science says. Explain how as a scientist you do your science. There will be people who, the most prominent voices out there, I never expect to have an effect there, because a lot of people have vested interests in spewing misinformation. People who want to claim that vaccines cause autism, lo and behold, they want to sell you an incredibly expensive, dangerous treatment for your autistic child. They have a vested interest. So they're not going to suddenly say like, oh, you're right. Yeah, thank you. I'm going to give up this healthy income stream. That's not going to happen. But, there are people that you could reasonably call anti-vaxxers, but there are lots of people, parents who might be called vaccine hesitant. Like, I don't understand vaccines very well. I've heard things that make me worried, and I don't want to do anything to harm my child. So, those people just want to understand what's going on. Now, if you sort of say like, well, any parent who would have any doubts about vaccines is an idiot, well, boop, you've lost them. So think about that. Think about, realize that you're in a human exchange. And I think that there is hope.
I bet you have some phenomenal stories to tell us about the science deniers who write back to you more and more these days. So here's another. You make a compelling case that race is a social construct, not microbiological. But most people don't know that, and genetics is a complicated science. How would you recommend that general education can change to help people understand that race is not a biological classification?
That's a great question. I mean, I think that, I am a big fan of, I'm a great admirer of high school science teachers, of grade school science teachers. Not only are they have this challenge of every day trying to go to work and make science exciting for teenagers and younger, but really I would say for the majority of people, that's probably going to be just about the last time that they are actually learning in class about science. And so, to help people to understand that, race is not a biological concept that has any validity, that it's historical baggage that really emerges out of the origin of slavery in the 1500s. Learning that in class is actually could be a great thing. And there have been some geneticists and high school teachers who have been trying to come up with how to teach this in high school. My colleague at the New York Times, Amy Harmon, wrote a really nice article a few months ago, kind of sort of seeing how it's going. I mean, it's a challenge because you're dealing with very hot button issues, and you may very well, discover that your students are getting all sorts of racist indoctrination at home, either explicitly or implicitly about, white people are just smarter than black people. That's just how it is. That kind of thinking. But it's a teachable moment. I mean, you can show people, well, what is human genetic variation? And we know what that is now. It's not just something that we're kind of guessing at. Like we have hundreds of thousands of genome sequenced from all over the world, and you know, skin color, which was so has such a charged history. Like, you know, it became such a marker for all sorts of toxic you know, racism. And we know about skin biology really well. We know like most of the genes that are involved with skin color. And, you know, a lot of the genes that, you know, I looked at my genome, and like one of the most powerful genes for why I'm so pale is present in lots of parts of Africa. It evolved, you know, about 200,000 years ago. So it's not like whiteness is some sort of distinct marker. In fact, you know, Europeans 10,000 years ago probably were not white, you know, in the sense of like, you know, what I look like. They were probably had quite dark brown skin, which, you know, white supremacists hate this, but, you know, that's just the facts. So I think there's great opportunity at the high school level or even earlier to get into these issues.
So this is our concluding question. Are shared family mannerisms inherited or acquired? Also, how far back can our cells hold ancestral DNA?
Well, so let's see. So I'll take that first question. So you know, your genes, you inherited those genes from your parents, and they inherited them from their parents. Now, along the way, generation to generation, a gene may have a very small chance of mutating. And so, but, you know, over millions of years, these genes have mutated a lot. You could just keep going back if you wanted, back and back and back and back. Really, you can go back all the way. So, you know, you and bacteria share a common ancestor, maybe 3 billion years ago. Life on Earth probably started about 4 billion years ago. It's the same genes, ultimately, that we all inherit, all living things, from those first life forms. It's just that those genes have mutated over time, and genes have duplicated and undergone more mutations. So really, like, you know, in terms of what you're inheriting in those cells of yours, you're inheriting the whole history of life on Earth. And so is every other living thing. But then you asked about sort of habits and so on that families have, and this sort of gets to the title of my book, She Has Her Mother's Laugh. You know, I mean, I hear phrases like that, you know, when we will talk about like, what did we get from our mother or our father or our grandparents or so on. And, you know, some people might talk about eye color, but other people will talk about laughter or the way we walk or you know, the kind of jobs that we hold and so on. And, you know, it's, for the most part, we're not talking about genes. And if we are talking about genes, we're talking about, you know, any one gene having an incredibly slight, subtle influence on different kinds of behavior. And so a lot of what you might assume has to do with genes is just the fact that, you know, maybe you grew up in a house listening to somebody laugh all the time, and that was just kind of how you learned to laugh. I mean, this is, you know, with birds, this is, you know, when birds learn songs, they're listening to the songs of the adult birds around them. They end up with almost exactly the same song in many species. But it's not because that song was in their genes. They learned it because it was what they were hearing around them. And then they pass it down to the next generation. That's totally inherited, but that's a cultural kind of inheritance. And what's interesting is that bird songs, certain kinds of birds, after many generations, the song changes. And so you might have the same species of bird, but living in different populations, and their songs will kind of split apart just because the song changes, you know. And if you're listening to a song later in the history of this population of birds, you're hearing a different kind of song, and you're going to pass that down to the next generation. And what's interesting is that, you know, if those birds come together, their songs are so different that, you know, the males and females may not be interested at all in each other, and so they won't mate. And so you might end up actually having two species evolving because of that cultural inheritance, not the genetic one. I'm not saying that we're going to turn into new species of humans because of how we laugh or the songs that we sing, but I will say that cultural inheritance is something we take for granted, and yet I think it really it has a huge influence on us for good and for bad. You know, we inherit, you know, we're dealing right now with a legacy of racism. We've inherited that culturally from hundreds of years of American history. That's a cultural inheritance. But, you know, we can change that. It's not written in our genes. So that's a more positive way of looking at what we've inherited, that we can alter it and then pass that down to the next generation.
That's such a good note to end on.
There you go.
I just want to say that if we were all able to unmute and applaud, it would be thunderous, and we just cannot thank you enough. And I'm going to hand this over to Kate.
I just want to thank Carl Zimmer for sharing your time and knowledge with us this evening, and Pam Ehrenkrantz and Laura Blum from the UJA-JCC Greenwich. We're so thankful for this collaboration. And finally, thanks to all of our participants this evening. We had some wonderful questions, and thanks so much for joining us. Hope to see you all soon.
Thanks very much.
Thank you all.
Thank you.